MRSA presence in high-traffic areas in the Health Professions Building at McLennan Community College. Presented by Hayden Durham. Mentored by Alyssa Petrie and John W. Seawright, PhD. Every surface around us holds millions of bacteria, even us. We unknowingly spread these bacteria around from our daily interactions. These bacteria are extremely beneficial in our day-to-day health, but can become harmful in certain circumstances. Not only can bacteria become resistant to treatment, but around 5% of healthcare workers can be carriers of these antibiotic-resistant bacteria. In this photo, we see a culture of E. coli. The little white discs that are spread around are antibiotic tablets. We can tell that some of the tablets have zero growth around them, while a couple have growth. This shows that this sample of E. coli has some antibiotic resistance. The antibiotic resistance that bacteria can develop are responsible for over 2.8 million antibiotic-resistant bacterial infections that occur in the US each year. Some of these infections are untreatable and lead to more than 35,000 deaths as a result. As we see here, this figure shows that certain antibiotic-resistant bacterial infections are increasing, while others appear to be decreasing or have no trend. The swab culture test was used in order to obtain these results in the hospitals. One particular bacteria, Methicillin-Resistant Staphylococcus aureus, the rates seem to be decreasing. Many bacteria, such as Staphylococcus aureus, are typical of the human microbiota. Staph aureus is also the most common cause for bacterial infection. This bacterium is round, shaped, and appears in chains and clusters. It is a gram-positive bacterium, which means it has thick layers of peptidoglycan within its cell walls. Like many bacteria, it can become resistant to antibiotics. Methicillin-resistant Staphylococcus aureus is an antibiotic-resistant bacteria that is resistant to beta-lactam antibiotics. Beta-lactam antibiotics are a classification of antibiotics that include penicillin, methicillin, amoxicillin, and more. These antibiotics induce cell death by producing a ring that disrupts steps in the biosynthesis of bacterial cell membranes. MRSA produces an enzyme known as beta-lactamase. Beta-lactamase breaks the beta-lactam ring that prevents the cell membrane's biosynthesis and therefore makes these bacteria resistant to beta-lactam antibiotics. In this figure here, we can see that nurses have the highest percentage out of the healthcare workers that are carriers of MRSA. Nasopharyngeal swabs were used to test for MRSA to find these results. However, it is estimated that around 5% of healthcare workers are carriers of MRSA. In the MCC enrollment profile for fall 2019, it is estimated that around 10% of MCC's total student population are involved in the healthcare program. Because of this, it is possible that MRSA is present in some places on campus. And given the prevalence of MRSA in healthcare settings, it can easily be transmitted from a healthy individual to a surface. We hypothesize that MRSA is present on the MCC campus and is more common in the Health Sciences Building, where student carriers are likely exposed to MRSA through their hospital rotations, in the student gym locker room, where students exercise, and in the MAC, where students congregate. Methods. To obtain each sample of bacteria, the swab culture test was used. This process includes using a sterile cotton swab with an application stick moistened in a sterile saline solution. From 4 locations, 3 different items will be tested for growth and for MRSA. These locations and items include the health simulation lab, the patient model, the bed rail, and the counter from the health simulation lab. From the MAC, the water fountain mouthpiece, a vending machine button, and an elevator button. From the gym, one free weight, and another free weight, and the weight bench. And from the male locker room, the door handle, the locker handle, and the shower handle. Once the samples have been collected, they will be transported back to the lab to be transferred to a blood agar plate and to an MRSA selective plate. The blood agar plate is composed of sheep blood to allow for growth of a wide range of bacteria. The MRSA select plate typically hinders the growth of bacteria that are not methicillin-resistant Staphylococcus aureus. The plates will then be incubated for 48 hours at 37 degrees Celsius, because this is the average human body temperature and the temperatures that Staph aureus grows best at. The health simulation lab was chosen because of the high exposure to health profession students. A swab will be taken from each item and then transferred to their own MRSA select plate and their own blood agar plate. From the sim lab, we will have 3 MRSA plates and 3 blood agar plates, a total of 6 plates. They will then be left to incubate for 48 hours at 37 degrees Celsius. The MAC was chosen because of the large body of students that frequently meet here. A swab will be taken from each item and transferred to their own MRSA select plate and a blood agar plate. From the MAC, you will have 3 MRSA plates and 3 blood agar plates, a total of 6 plates. They will then be left to incubate for 48 hours at 37 degrees Celsius. The gym. The gym was chosen because MRSA is more likely to be found in athletic facilities. A swab will be taken from each item and then transferred to their own MRSA select plate. From the gym, we will have 3 MRSA plates and 3 blood agar plates as well, a total of 6 plates. They will then be left to incubate for 48 hours at 37 degrees Celsius. From the male locker room, which was chosen because MRSA is also more likely to be present here, such as in athletic facilities, we will have 6 plates total. We'll have 3 MRSA plates and 3 blood agar plates. They will then be left to incubate for 48 hours at 37 degrees Celsius. It's now 48 hours later. We will view the plates, take pictures, and record any growth. Blood agar plate growth shows that bacteria are present. We will check the MRSA plates to determine if there is any growth. The MRSA plates with no growth were deemed negative for MRSA. Once we viewed the plates and we found MRSA, we gram-stained it. When gram-staining, you are testing for whether a bacterium has a thick or thin peptidoglycan layer. Gram-positive bacteria have thick layers of peptidoglycan within their cell walls. This means they hold the crystal violet stain well and will appear a dark purple. Gram-negative bacteria have thin layers of peptidoglycan, so they do not hold on to the crystal violet well. Instead, they wash it out, which allows the safranin dye to show through and will appear pink. Determining whether or not a bacteria is gram-positive or gram-negative will help in determining which antibiotic to use to treat it. In order to gram-stain, you must apply the crystal violet and wait a minute, rinse with DI water, apply grams iodine, wait a minute, rinse with DY water, rinse with alcohol solution, apply safranin and wait 1 minute, rinse with DI water, and then view under a microscope to find out if the specimen is gram-negative or gram-positive. Results. In the health simulation lab, only the counter's blood agar plate showed growth. None of the Mrsa plates showed growth. In the MAC, only the vending machine button and the elevator button showed blood agar growth. None of the MRSA plates showed growth. In the gym, only the second free weight and the weight bench showed growth. None of the MRSA plates showed growth. And in the male locker room, all 3 blood agar plates showed growth, but only the shower handle showed MRSA growth. MRSA spots. The pink spots show that MRSA is present from the shower handle. The light pink spots surrounded by non-pink spots, which are most likely forms of bacillus that are not hindered by the specialized agar growth. Gram-stained MRSA. Clusters of Staphylococcus Aureus represent the MRSA. The violet represents that it is a gram-positive bacteria. Results. 7 blood agar plates had growth. 11 MRSA plates showed no growth, and 1 MRSA plate showed growth. Conclusion. It is not shocking to find MRSA in the male locker room. Because MRSA is more likely to be found in athletic facilities, it would not come as a shock to find it in the female locker room as well. This is not a healthcare setting, but instead is a frequently used and shared item. This could be due to the high incidence of exposure. And unlike the gym, which has self-sanitation policies, and the MAC and Health Sim Lab, which have increased sanitation techniques, the shower handle is not cleaned after every use. The shower often reaches a temperature hot enough that the bacteria prefer to grow in. One solution to make sure MRSA does not remain here is to have users spray down the high-touch areas with a disinfectant solution after each use. Acknowledgements. McLennan Community College Honors College, John W. Seawright, PhD, Elisa Petrie, John Spano, PhD.